This is the fourth presentation in the series about the biological basis of learning disorders. This presentation looks at autistic spectrum disorder, ASD. ASD is a spectrum. That is to say, there's a range of symptoms or characteristics ranging from those with severe social and communication and interaction problems to those with relatively mild conditions who can function normally in society. ASD normally affects people in two different ways: their social communication and their social interactions. People with ASD have difficulties interpreting both verbal and non-verbal language, like gestures or tone of voice. Some with the more severe conditions are unable to speak or have very limited speak, while others have good language skills but struggle to understand sarcasm or tone of voice. People with ASD often have difficulty reading other people, recognizing their feelings or intentions, or expressing their own emotions. This can make it difficult for them to navigate the social world. People with ASD may appear to be insensitive. They may want to be on their own when overloaded by the demands of other people. They may not seek comfort from other people and may appear eccentric or strange or behave in ways that are socially inappropriate. And as a result, they find it hard to form friendships. Autism spectrum disorder is a relatively common condition, with about 1 in 88 children displaying some form of characteristics, although of varying degrees of severity. It's a neurodevelopmental condition and it has a highly heritable component. Somewhere between 50 to 60% of the differences observed are caused by genetic factors. The more severe aspects of ASD are often caused by quite specific chromosomal genetic abnormalities, such as Prader-Willi syndrome or Fragile X. Genome analyses of people with ASD symptoms show a large number of genetic variants associated with the condition. There's nearly 3 million distributed across all 23 pairs of chromosomes, which seem to be associated with the condition, and they are inherited from parents through to their children. We're becoming increasingly rare that the condition can be affected by relatively rare variants that very few people in the population have, and as of this moment, about 300,000 of those rare variants have been described across the world. Sometimes children develop mutations inside their bodies that their parents do not have, and there's about 70 mutations which are described as being de novo have arisen within the children. Some of these could well be passed on to their children's children. Many of the genetic variants described on the previous slide occur in the non-coding regions of the genome and therefore do not directly affect protein production. But there are many genes which are now associated with a susceptibility to ASD, which have been found on almost every chromosome in the genome. A number of the most important ones are listed below with their chromosome number. These genetic factors and these protein coding genes affect the development of the brain, and therefore, as this slide shows, are most likely to be active during embryonic development and in the first few months and years following birth. The brain organises itself during childhood and adolescence, and therefore some of these genes may well be operating well into the teenage years. There have been a number of genomic studies of ASD over the years, and they're beginning to paint a consistent story of a large number of mutations having relatively small effects upon the development of the condition. Some alleles seem to increase the level of risk by about 6-fold, but each one is relatively small. There seems to be about 500 protein coding genes which to a greater or lesser extent contribute to ASD in the population. These alleles are inherited from parents, and there appears to be effect of paternal age of the father, on the mutation rate and the incidence of ASD. So older fathers are more likely to pass on some of these alleles to their offspring than younger fathers. A key factor in all of these genomic studies are the idea of copy number variations, which are pieces of DNA which are duplicated across the genome and exert a greater effect because they are present in greater numbers, or deletions of genome bases which alter the functioning of the normal DNA. Again, these are inherited from parents during sperm and egg production. One particularly severe form of ASD is known as Fragile X syndrome, which is caused by a mutation in a single gene known as FMR1. FMR1 produces a protein called Fragile X protein, which is involved in the regulation of the production of hundreds of proteins that are important for brain development, for aging, and for synaptic plasticity, which is how nerve cells communicate with each other. When someone has a full mutation of FMR1, usually boys, there is very little protein produced, leading to severe symptoms of ASD.